All right, today we are diving deep into the world of carbohydrates. And trust me, they are so much more than just what's in your pasta and bread. These molecules are kind of like the hidden architects of the entire biological world. Their energy, their building blocks, and they're even the unique ID tags on our cells. Let's get into it.
Okay, let's kick things off with a little puzzle. What on earth could possibly connect the energy you get from a loaf of bread, the super tough shell of an insect, and the A, B, or O marker that determines your blood type? I know, it sounds totally random, right? But the answer that ties all of them together is, you guessed it, carbohydrates. So, to really understand their incredible power and just how diverse they are, we've got to start at square one. We're talking about the simplest units, the monosaccharides. Think of them as the fundamental building blocks for literally all other carbohydrates.
You know, the name carbohydrate itself gives us a massive clue about what they are. It literally means a hydrate of carbon, and that's because for a lot of these simple sugars, the chemical formula basically looks like you have one carbon atom for every one molecule of water. Now, of course, there are exceptions. Deoxyribose, the sugar in our DNA, is famously missing an oxygen atom, which is why it has that deoxy prefix. But hey, the general name stuck.
What's really cool is how logically we name these sugars. It's actually a pretty simple, elegant system. First, you've got a prefix that tells you how many carbons are in it. Tri for 3, pent for 5, hex for 6. Easy enough. Then, another prefix tells you its main chemical group. Aldo if it has an aldehyde, or keto for a ketone. So, a sugar like glucose, which we all know has 6 carbons and an aldehyde group, is technically called an aldohexose. The name literally tells you its blueprint.
Okay, but here's where things get really fascinating. The chemical formula, C6H12O6, doesn't even begin to tell the whole story. It's the three-dimensional arrangement of those atoms, the actual shape of the molecule, that changes everything. It's how you can get dozens of totally different sugars from the exact same set of atoms.
And that brings us to this key concept, diastereomers. These are molecules that have the same chemical formula, but they aren't mirror images of each other. The only thing that's different is the 3D orientation of their little hydroxyl groups. So you take glucose, galactose, and mannose, they all share that same C6H12O6 formula, but they are completely different molecules with totally different jobs in the body, all because of these tiny shifts in shape.
Now let's zoom in even closer on a special type of diastereomer called an epimer. Think of these guys as almost identical twins. Their chemical structure is exactly the same except for the orientation of atoms around one single carbon.
And this is the perfect example. Look at glucose and galactose. They are epimers. The only difference between these two sugars is which way the hydroxyl group is pointing on carbon number 4. That's it. And that one tiny change has huge biological consequences, and it completely changes how our bodies can use them.
So, what if instead of flipping just one of those hydroxyl groups, you flip the orientation of every single one? Well, then you get a perfect, non-superimposable mirror image. These pairs are called enantiomers. It's just like your left and right hand. They're mirror images, but you can't stack them perfectly on top of each other. D-glucose is the form our body loves and uses for energy, but its mirror image, L-glucose, our body can't use it.
But here's a really crucial detail. In the watery environment inside our cells, sugars like glucose almost never stay in those long, straight chains we draw on paper. They have this natural tendency to curl up on themselves and form a much more stable ring structure. So, what happens when they do that?
Well, this act of forming a ring actually creates a brand new special carbon that wasn't there before. We call it the anomeric carbon. And depending on whether the new hydroxyl group on this carbon points down or up, we get 2 new versions called anomers, the alpha form and the beta form. And believe it or not, this simple little distinction is literally the difference between starch, which is a source of energy, and cellulose, which is an indigestible fiber.
Oh, and these rings aren't flat like a washer either. To relieve all the internal strain from the atoms bumping into each other, they pucker up into 3D shapes. The 2 most famous are called the boat and chair shapes, and the chair form is usually way more stable, because it lets all those bulky hydroxyl groups spread out and have some personal space, which minimizes all that chemical stress.
Okay, so now we have our basic sugar units, and we know they come in all these different shapes. So let's see how we can start modifying them and snapping them together, kind of like Legos, to build much bigger and way more functional molecules. So a really key chemical property of a sugar is whether it's reducing or non-reducing. And it all comes back to that special anomeric carbon we were just talking about.
If its hydroxyl group is free, the ring can actually pop open back into its straight chain form. This exposes a reactive group that can donate electrons, making it a reducing sugar. And we can actually test for this in the lab with something called Benedict's test, which turns from blue to orange if a reducing sugar is there.
So when sugars link up with each other, they form this really strong covalent bond called a glycosidic bond. This happens when the anomeric carbon of one sugar reacts with a hydroxyl group on another molecule. And once that bond forms, it's like a lock. That sugar is now stuck in its specific alpha or beta form for good.
And this is exactly how we get the disaccharides that we all know. Sucrose, regular table sugar, that's just a glucose linked to a fructose. Lactose, the sugar in milk, is a galactose linked to a glucose. And maltose is just two glucose units stuck together. Simple as that. Now we're getting to one of the most important roles that carbohydrates play. When you attach these complex sugar chains to proteins, you create something called a glycoprotein. And these sugar chains, they act like these super sophisticated molecular identifiers on the surface of our cells, kind of like a cellular barcode. But right here, we have to make a super important distinction between two processes that sound almost the same but are worlds apart. On one hand, you have glycosylation. This is a deliberate, enzyme-controlled, precise process of attaching sugars to proteins. It's essential. On the other hand, you have glycation. This is just a random, messy reaction that happens when sugars bump into proteins by accident. This uncontrolled process can be really damaging and is linked to things like aging and diabetes. It's actually the exact same reaction that makes your toast turn brown.
The most common type of that good, controlled process is N-linked glycosylation. And you can think of it like a three-step assembly line. First, a core sugar structure gets built on a lipid carrier. Second, this whole thing gets flipped into a different part of the cell, the ER, where the sugar chain is finished. And finally, this entire complete sugar tree is transferred as one big unit and attached to the target protein. It's incredibly precise. Okay, so we've looked at single sugars and pairs of sugars. But what happens when you link together not just two, but thousands of them? Well, then you create polysaccharides, the true heavyweights of the carbohydrate world. And they're used for both massive energy storage and incredible structural strength.
This slide just illustrates the point perfectly. It shows how one tiny little chemical detail can change absolutely everything. On the left, amylose, which is in starch. On the right, cellulose, which makes up plant walls. Both are just long chains of glucose. The only difference is the bond. Amylose uses alpha-1,4 bonds, which makes it form a nice spiral shape our enzymes can easily break down for energy. Cellulose uses beta-1,4 bonds, which makes it form straight, rigid fibers that we can't digest at all. That one little flip from alpha to beta is what turns food into fiber.
And cellulose isn't the only structural superstar out there. Remember the bug shells from our puzzle at the very beginning? That's all thanks to a polysaccharide called chitin. It's a lot like cellulose, but it's made from slightly modified glucose units. And this little change allows the chains to bond even more strongly, creating the incredibly tough material that forms insect exoskeletons and even the cell walls of mushrooms. All right, let's bring it all home now and look at how all these principles of complex carbohydrates show up in the world all around us, in both biology and in technology.
I mean, they're just everywhere. Viruses like the flu use special proteins called lectins to grab onto specific sugars on our cells to infect them. We can break down the tough cellulose in plants to get glucose from making biofuels. In the science lab, a polysaccharide from seaweed called agarose is what makes up the gels we use to separate DNA. And inside our own bodies, long polymers like hyaluronic acid act as super important lubricants in our joints.
So it's pretty clear that carbohydrates are speaking this complex, versatile chemical language all around us and inside of us. They are so much more than just simple fuel. They're master builders, they're cellular communicators, and they're the gatekeepers of our biological identity. From a single simple sugar ring to the complex code covering every one of our cells, they are absolutely essential to life. Which kind of leaves us with one final thought. What hidden messages are your body sending with its sugars today?